I'm going to take the next 13 minutes or so and introduce you to the company that I lead, StemBioSys. And what I want to start with, let's see if I can, there we go. What I want to start with is where we started the company. Our scientific founder, Dr. Chen, was working in his lab about a decade ago, and he was working on expanding bone marrow MSCs. And the challenge he kept running into is when you took the MSCs out of their environment and isolated them, the state of technology at the time, when he put it into a dish or he put it into some other substrate, the cells did things he didn't want them to. They started to differentiate, they started to change phenotype, and what he wanted to do was get a large number of high-quality MSCs that could support his research. So he stepped back and he said, well, what if I take the cells out of their native environment, I isolate them, and I come up with a way to develop in vitro a model that looks very much like the cells we're used to when they were still in vivo. And in fact, that's just what he did. He took a process. He started with bone marrow MSCs in this case. He seeded them into a plastic dish. He added media and growth factors, got the cells to lay down an extracellular matrix, and at the end of the process, he gently removed the cells, leaving the extracellular matrix. And what he found is that when he put bone marrow MSCs on that matrix, they retained their phenotype. He got more cells than he did on competing technologies. And when he started to look at genetic markers, things like stage-specific embryonic antigen 4, he got a lot more expression of that. And so he said, you know, I think I'm onto something. And in fact, he and another San Antonio doctor got together and actually put together a company. I stepped in about 5 years ago and became the CEO of StemBioSys, and we took this technology and we said, so what are we going to go do? And I'm going to take you through sort of the story of how we've built out this company and what we've done. And I'll start with the strategy that we followed. What we've put together is a three-pronged strategy for the company. The first is we said this is a research product that we believe would have applicability to researchers and pharmaceutical companies around the world in various kinds of research. Two, we said we don't want to be arrogant about this. This is a platform technology at the end of the day, and the highest and best use of this technology may in fact get discovered outside of the four walls of StemBioSys. And so we set a very aggressive program to identify collaborators around the world that we could work with where this technology might be helpful to the work they were doing. And I'll share with you some of the data on that. And we had a long discussion about therapies and what we were going to do about therapies. And there was even some discussion early on in the history of the company, should we become a stem cell therapy company? And what we have concluded is that there is a lot of potential out there, but for purposes and to keep fundraising needs to a minimum, we were going to focus only on early feasibility study to give us data that would then allow us to apply for government grants. And in fact, that's exactly what we've done, and we've had a couple of grants, and I'll share with you some of the data that's come out of some of that work. So let me start with the life science tool business. And if you look at the chart on the top, the very first product we had, we called our Salvo matrix. So the tagline we had was the next evolution in cell culture. We took that product out to market, and the very first thing we heard is that it's a great product, but you use fetal bovine serum in it, and there were customers who wanted a xeno-free version, so we developed a xeno-free version by replacing the fetal bovine serum with human platelet lysate. That was our second product. We also realized that this was really good at expanding cells, and we said, well, wait a minute, there's probably a market for us if we were to go into the cell market, and in fact, we have now 5 cell products that we have launched, and we also launched a media product. We discovered we were sending our customers, to other media companies, and that felt silly, so we wanted to offer a complete set of solutions. It's interesting as the company has evolved in this life science tool space. Right now, about 70% of the company's revenues are coming from the sale of cells, and that's actually been, for us, where we've gotten a lot of traction in the commercial space. So researchers tend to buy our matrix in an academic setting, but big pharma companies are buying our cells. It's been an interesting process. Slide 7 just highlights sort of the journey we've been on from a product development perspective. You can see where we were in January of this year, 2 matrix products, 6 cell products, and as we look toward the end of this year, we'll have doubled the number of matrices, and I'll talk about each of those in a second. We'll add 2 cell lines, and we have launched our first media product over the course of the summer. As we look forward to 2020, again, lots of additional product development coming. You can see the work that we've got going there. Products are important, but without the right channels to market, you have absolutely nothing, and you don't have a business. And one of the things we've done is to begin to build our own internal sales capability. We hired our first chief sales officer a year ago. We also recognized that we could not do this alone, and that what we needed were a set of good partners to bring these products to market. And in fact, we have set out to do just that, and we have now 3 distributors here in the United States, including my friends at Gemini Bioproducts who are here in the room. VWR Thermo Fisher, we have a great distribution partner in Europe. We've got a distributor in Japan, 2 in South Korea, 1 in Singapore, and a distributor in Australia. And we have just launched a discussion with a potential distributor in Israel. The one market we continue to struggle with is China. We have had a number of direct sales to the Chinese market, and the question just is what kind of partnership we want to do, and that's something we're continuing to look at. So that's the life science tool market, and let me now begin to take some of the covers, if you will, off of 2 of the collaborations that we have worked on. One is with the University of Michigan, and that's been a really interesting piece of work that has actually been going on for 2 years. The Michigan work, we developed a novel form of our matrix. It is not available yet on the market. It was something that we did together with them. And they identified a very interesting application with regard to cardiac cells, in particular cardiomyocytes. And there may be some really interesting potential applications around cardiac toxicity testing and cardiac disease modeling. We partnered with Michigan about a year ago to put together a phase 2 SBIR grant, and in fact that grant was awarded. It was a $1.3 million grant that we received earlier this year, and the teams on both sides are off and running on that, so we're very excited about that. There will be a paper published in the first half of next year on the data on this, so stay tuned for more details on the work at Michigan. And then the work at Rutgers, this was with Joachim Cohen's lab in the New Jersey Center for Biomaterials. Joachim, we had a discussion about chondrocytes, and there's a lot of challenges with expansion of chondrocytes. Those of you who are familiar with this or have worked with them know these are cells that de-differentiate pretty quickly as you put them into as you try to expand them. And so we asked ourselves the question, could we take the technology we have, go back to that first slide where you think about I started with bone marrow MSCs, and we asked ourselves, well, what if we started with chondrocytes and had chondrocytes make a matrix and go forward from that? What would we see? And in fact, that was the work we did with Joachim late last year. And what we found across the top is that, in fact, you get better chondrocytes. So what we looked at here, we put chondrocytes on tissue culture plastic, we put chondrocytes on our bone marrow matrix, and we put chondrocytes on the special chondrocyte matrix that we made for Dr. Cohen. And we saw, in fact, if you look at expressions of collagen 2 relative to collagen 1, you saw higher level of collagen 2 on the cells expanded on the chondrocyte matrix than you did on either of the other 2. Better cells are great, but if you can't get enough of them, you don't really have anything. And in fact, what we saw was we got more cells on that chondrocyte matrix. So we were very excited about this. This data was actually published in January of this year, and we are currently applying for phase 1 SBIR grant to take this to the next logical step, which would be a small animal model. And if that's successful, obviously in a phase 2, we'd want to go to a large animal model to determine whether or not these better cells, in fact, translate to better clinical outcomes in terms of cartilage growth. So again, stay tuned on that one. We have 2 products that we are going to be launching this quarter that we want to share with you today. I've talked a lot about cells, and if you looked at our cell map, you saw a lot of human-based cells, adipose cells, MSCs, et cetera. The one thing I haven't talked about is induced pluripotent stem cells, and that is because until today, we really didn't have a solution for induced pluripotent stem cells. But I'm pleased to announce that this quarter, we will launch our Salvo Matrix Plus product. It is directed specifically at induced pluripotent stem cells, has the benefit of being as good as, at least, the gold standard products that are out there around the expansion of iPS cells. as well as you've got a really interesting biological relevance because this is derived from a human cell, so all of the biological relevance that you look for in our matrix products, it has. We've also seen some very interesting early data that seems to indicate that when you put iPS cells on this and differentiate them into another cell, through whatever protocol you're using, the cells actually will evolve into a mature phenotype. And many of you know that one of the challenges with iPS cells is they retain a fetal phenotype when you differentiate them, and this may be part of the solution that's out there. And a final comment I'll make, this is an extremely easy to use product. It can be kept refrigerated. It's about a 45-minute to an hour-long process to rehydrate it, and then it's ready to go. So a lot of the challenges associated with some of the competing technologies here don't exist. So stay tuned. We'll have that product out, as I say, by the end of the year. I've talked about on the left-hand side there the chondrocyte matrix. That product will also be out this quarter. We've actually shipped that product to a customer in Europe. We warned them. We said, you're getting the first production run. Be careful. And they said, no, we're willing to try. So in fact, we had a call with them on Monday, and they were very happy with the results they had saw. So that was great. We've also begun work on a neuronal matrix. Very interesting early results. We've been working with a researcher locally in San Antonio who does work with this, and I anticipate that we'll have that product on the market by the middle of next year. So I've talked a lot about the products, where the company is, where we're going. The one thing I have not talked a lot about yet is the team that we have behind it. I'm very privileged to be part of a board of just outstanding people. Our chairman, Kathy Berzig, has had a long, illustrious career in a variety of medical and healthcare companies. Those of you familiar with AdvaMed may be aware she received their Lifetime Achievement Award last week. So we were very happy for Kathy on that. We have a number of people from the San Antonio business community with backgrounds in the pharmaceutical space, members from the venture community, and then to provide us the appropriate level of scientific perspective on this, Bob Langer at MIT is also on our board, and I always get the question, is Bob involved? And Bob is deeply involved and holds us all accountable, I assure you. So that's StemBioSys in a quick nutshell. Again, great life science tool business that's off and running. We've had some interesting collaborations, some interesting early therapeutic data that we're beginning to look at, and we're very excited about the year ahead. Thank you.